Hello, my name is Mark Anthony. I'm a third year PhD student at the University of Surrey. So recently I was privileged to receive IBNET's travel bursary to attend the 10th IWA Microbial Ecology and Water Engineering Specialist Conference in Brisbane, Australia. So I gave a platform presentation at the conference, and I must say it was indeed a rewarding and amazing experience, as I got to meet and network with peers working in similar research area. And most importantly, I got to attach faces to some of the wonderful and admirable authors whose work I have read and cited in the course of my PhD journey. So what I'd like to do in this video is to give a summary of my platform presentation at the conference. So my PhD project is focused on optimizing the anaerobic digestion of farm waste to mitigate the risk of disseminating antimicrobial resistance via the application of digestates to agricultural land. And we do know that surveillance and monitoring are key components of strategies intended to combat the environmental dissemination of AMR. So keeping that in mind, what we did was to sample the feedstock and digestate of 5 commercial biogas plants operating between 41 and 44 degrees Celsius. So 2 of the sites we sampled feed exclusively plant-based substrates, while the rest of the sites feed a blend of purpose-grown crops, manure, as well as brewery waste. So we extracted DNA from the samples we collected and then proceeded to conduct shotgun metagenomic sequencing. The findings were pretty amazing and interesting. So we found a total of 160 subtypes of antibiotic resistant genes in the samples, and the distribution of the resistant class was quite reflective of the antibiotic use and prescription in human and animal medicine in the UK. So some other thing we found was that there was significantly higher richness and abundance of antibiotic resistant genes in the sites that feed animal manure. So the total abundance of antibiotic resistant genes actually decreased by 49 to 92% after primary digestion, with higher rate of reduction observed at sites operating at higher temperature, that is sites operating at 44 degrees Celsius. So putting our findings together, we made some keynotes. So firstly, the substrate type and temperature are key determinants of resistome trends in anaerobic digestion. Secondly, we noted that the hyper-mesophilic temperature, so we defined the hyper-mesophilic temperature as temperatures between 40 and 50 degrees Celsius. So this temperature, we believe, can achieve up to 92% reduction in abundance of antibiotic resistant genes. However, some high-risk genes may persist in the digestate. So another note we made was that human-associated pathogens were a repetitive host of multiple ARGs in the digestate we sampled. So lastly, we recommend that the contributions of plant-based substrate to the environmental burden of antibiotic resistance should be given more attention. So that's about all I presented at the conference. So see you in the next one. Goodbye.